talking to you. This is Goddess of Science Biology, and I'm Mrs. Bolden. Today we're going to talk about the light-independent reactions in part 6 of the cellular energy series. Okay, so we talked about the first two parts of the process of photosynthesis. This is the third part, which is the light-independent reactions, although it's kind of a misnomer because the light-independent reactions actually depend upon the products of the light-dependent reactions to happen. So it's a little bit of a misnomer, but we're going to go with it. We're not up to changing that these days. It's also called carbon fixation or the Calvin cycle. Guess who discovered it? Yep, it was Calvin. So carbon dioxide comes into the cycle because plants and other autotrophs take in carbon dioxide. And that immediately hooks up with a 5-carbon molecule. And on the next slide, I'll show you kind of the flow of what's going on here. You just kind of have to bear with me until you can see this for yourself. That then falls apart into 2 molecules that have 3 carbons each. So we had 5 carbons to start with that was already in the cycle. We added another carbon, so that's 6 carbons. That molecule falls apart into 2 3-carbon molecules of phosphoglyceraldehyde, or PGA. Just remember that the carbon dioxide with the 5 that's already in the cycle makes a 6-carbon thing, which falls apart into 2 3-carbon things. So all of our carbons are accounted for. So if you have 6 carbon dioxides, you're going to end up with 12 PGA coming out the other end. So 10 continue on in the cycle, and you end up with those 10 regenerating the starting 5-carbon molecule that hooked up initially with the carbon dioxide that was entering. 2 leave as one of those alphabet compounds, G3P, or if you want to use the whole name is glyceraldehyde 3-phosphate, and those 2 go off to make glucose. Yay, we're finally making some glucose here. So here's kind of an overview of what's going on here. Here's our carbon dioxide that are coming in. They hook up with this 5-carbon thing that we already have and make 12 molecules of 3-PGA. Little ATP, 12 ATP, get turned into 12 ADP. And we have some NADPH, which gets the hydrogen stripped off of them. And so we end up with 12 3-carbon molecules, which most of them go back and make this 5-carbon thing that we're going to need to continue the cycle. But 2 of those 3-carbon molecules end up making glucose, which is a big yay in this whole thing, because that's what we want to do to start with. Now, sometimes conditions are rather harsh in order for photosynthesis to take place because, as you know, reactions oftentimes are temperature dependent. So if you have greater than 77 degrees Fahrenheit or 25 degrees Celsius, the Calvin cycle has a little burp in it here. It makes more carbon dioxide and less glucose, which is not really good because we're only getting 2 glucose out of this thing. So in order to reduce, plants often to reduce the loss of water, they're going to close their stomata, which are little tiny pores that are in the surface of the water, but they're kind of like eyelids, and they can open up and then water and other gases can leave. Also, new gases can come in. If it's on a really, really hot day, to reduce the water loss, plants just automatically close their stomata or their pores. So it kind of cuts off the carbon dioxide supply because the carbon dioxide is coming in. That's a problem because this whole thing, the Calvin cycle, all has to have carbon dioxide to run. So we have a problem. So obviously there are plants that exist in environments that are greater than 25 degrees Celsius or 77 degrees Fahrenheit. So there must be some ways around that. So we have a couple of alternative pathways that plants use. So normally, C4 plants, this is the first one. C4 plants, some examples are sugarcane, corn, sorghum, other grasses. All of those things are grasses. Even though you wouldn't be planting your front lawn with a whole, all the sugarcane that you can put in there, they're still in the same family. So what they do, C4 plants, is instead of, remember back in the Calvin cycle, the carbon dioxide is fixed into 2, 3 carbon compounds. Well, now they're going to fix the carbon dioxide into a 4 carbon compound in the Calvin cycle. And what that does is that reduces the need for as much carbon dioxide as the regular Calvin cycle happens. There's not enough carbon dioxide to keep the Calvin cycle going on a regular basis, so we have to do it some other way. The other way that this happens is the other solution to the we need to photosynthesize even at high temperatures is something called the CAM plant. CAM plants are crassulacean acid metabolism, and you wonder why they call them CAM plants. So this is a different kind of photosynthesis. So this occurs in real water conserving plants, things that you traditionally think about as plants that live in very desert conditions, things like that. Cactuses, sedums, pineapple, other desert plants. Those are usually CAM plants. So the stomata open up at night and close during the day. Well, that kind of makes sense because plants want to conserve their water. But the stomata at night is going to let the carbon dioxide come in at the nighttime. It's usually cooler and more humid at night, so we're going to be able to take in more carbon dioxide. So the carbon dioxide fixation in the CAM plants only occurs at night because that's the only time that we have any carbon dioxide available. So the fixed carbon dioxide gets released during the day into the Calvin cycle, and that's it. Thanks for joining me. Have a great day.